Right, so remember, what two domains contain prokaryotes? Bacteria and Archaia. So they've actually divided them to two separate chapters in your book now, where in the past they were all together as one. Although we know the most about bacteria, eukaryotes are relatively new on the scene for us. Excuse me, Archaia. I said I could do this in my sleep, didn't I? Apparently I was wrong. Eukarya, we're learning more and more, right? They're relatively new to us as far as microbiology goes. So most of my lecture and most of what we talk about really does concentrate on bacteria. But we will talk about the differences between, some of the subtle differences between bacteria and Archaia as we go through this prokaryotic lecture. So for especially bacteria, they have various sizes and shapes. And for the shapes, we have lots of different terminology that we use to describe the most commonly seen shapes. Now in microbiology lab, it gets about as exciting as the first two. We pretty much only see cocci and bacilli. Spirillum, I have some prepared slides, but we don't have any organisms that we grow that are spiral shaped. Mostly because it's too difficult to grow them. They're a little bit picky. Triangular and square are some of the shapes newer shapes that we see with Archaia. But most of us know what a triangle and a square looks like, right? But you're probably still wondering what the heck does she mean when she says coccus and bacillus. So we're going to take a look at those. And then sometimes the cells, when they divide, they stay next to each other. Now, even though there's individual free-living organisms, right, a single cell is a single organism when we talk about prokaryotes. They're unicellular. But some of them will stay next to each other when they divide. And they'll form these characteristic arrangements, or what we sometimes refer to as groupings. So you've probably heard of streptococci before, right? Because Streptococcus prurigens, anyone know what infection that causes? Strep throat, right? And so the name that we give to that is just an abbreviation, actually, of the name of the organism that causes that infection. This particular one, strepto, actually refers to chains. And so each part of these refers to, again, what would you see if you looked? If you looked under the microscope, you'd see they form a chain. They stay in a line. So let's look at some of these pictures. So coccus, or cocci, if you're talking about more than one, are spherical. So three-dimensional organisms, remember, right? We're not talking about circles here, right? It's like a ball, like a globe. It's spherical. Coccus is what you would say if you're talking about a single cell. It's a coccus. Cocci is what you would say if you're talking about more than one cells that are spherical in shape. So this is Latin. Anyone familiar with Latin? So I is actually plural in Latin. And us is singular, right? That ending. So I always think of it as opposite than what we're used to in the English language, right? Because when we say I, we're talking about ourselves, just a single being. But in this case, I is plural. And when we say us, we're talking about a group, right? Like this class. But in that case, it's singular, right? When we say coccus, we're actually talking about a single cell. So if you remember that it's opposite than what we're used to, then usually you don't get confused. Now, rod-shaped, and a lot of scientists say rod instead of the correct term, which is a bacillus. And again, these are three-dimensional organisms. So this rod is almost like a hot dog, right? Or a hot tamale, one of my favorite candies I probably should have stuck with the other day. Instead of the Reese's Pieces. So again, bacillus is singular if we're talking about just one. Bacilli is plural. Now, another reason why scientists like myself, a lot of times instead of saying bacillus, we say rod-shaped, is because bacillus is actually also a genus name. Right? So, yeah, I know, typo. genus name. A species would be one that we've learned about. If I could spell today. I think that one is CIS. Anthraxis. Anthraxis. I should know this. My brain does not want to work. Ha! I got it right. Save. So as you can see, it's somewhat confusing, right? If somebody just said bacillus, you wouldn't know whether they were talking about the genus or the shape. The good news is that genus, guess how they got that name? By their shape, right? They're all rod-shaped. So that makes it easy. But how would you know that somebody was talking about the shape as opposed to the genus? Right, the context of the conversation would hopefully tell you. There's also one quick clue that would clue you in that they're actually talking about the genus as opposed to just the shape. Notice how we write the genus. We italicize it. And if you handwrite it, you would underline it. We always capitalize the genus name too, and we never capitalize the species name. It's always lowercase. But again, that wouldn't right away tell you because you capitalize any letter at the beginning of a sentence, right? But that italicized would really indicate to you right away that they're talking about a genus as opposed to just the shape. Make sense? And again, why we just make it easier and we just say rod-shaped usually, right? Instead of saying bacillus. And of course my animations are backwards. Lovely. Okay, I probably fixed this PowerPoint somewhere. So these I have to say I've never really seen for sure. There are apparently some that are really short rods that almost even look oval shaped. And so because they're such short rods that sometimes they almost even look like cocci, because remember, they're three-dimensional organisms. So imagine a hot dog, right? Everyone knows what a hot dog looks like. If you look at a hot dog the long ways, right, you can definitely see it's almost like a rod. But if you were to turn a hot dog and look at it long ways, right, at the very top, it would look almost like a circle, wouldn't it? You guys visualize that? Right? So almost like taking your water bottle and looking at the top of it, right? It'll look circular. So remember, you know, in three-dimensional space, depending on how they're oriented, they may kind of look, because they're such small rods, they may kind of look spherical, right? But they're not. And in microbiology lab, we only have cocci or rods. So I always tell my students with the little rods, I'm like, is it a perfect circle? They're like, no. I'm like, then it's a rod. Right? If it's not a perfect circle, right, if you're not seeing a perfect sphere, then you're looking at a really small rod. Although most of us don't use the term coccobacillus, but it is out there. Vibrio, on the other hand, we do use that term. And again, this looks like a rod, but it has a curve to it, one little curve. Just like bacillus, this one is also a genus name. So as you can imagine, all the organisms in this genus look like this. They look like little rods with a little curve to them, almost like commas. So Vibrio cholera is an example of an organism that has this shape. And of course it causes cholera. Also note in this picture, you see this little line right here? And this is an electron micrograph. You wouldn't see it in this much detail under a light microscope. It has a flagella, right? Most of the organisms in this genus have this shape, and you have flagella. They are motile. The next would be Spirillum if you're talking about just one, or Spirilia if you're talking about more than one. And these guys have multiple curves to them, so it's like a really long rod, right, with multiple curves. Most of us know what spiral is like. And again, in this picture, notice these thread-like things. These are flagella, multiple flagella at both ends of this organism, at both ends. The last one are spirochete, or spirochetes, or spirochete, some people say. These guys are helical, which means three-dimensionally they look like a slinky, right? Or a spiral staircase. They have this particular shape because they have axial filaments. These are similar to flagella, but they run along the outside of the cell within an outer sheath. And so these black lines that you see here are those axial filaments. And they've actually genetically engineered spirochetes without the ability to make axial filaments. And they just end up being this long rod. They can't form that characteristic helical shape without the axial filaments. So those filaments actually contribute to this helical shape that you see for these organisms. And their unique motility. They actually corkscrew through their environment, right? Through mucoid or liquidy-ish environments, they can actually corkscrew through. One of the diseases that we listed out of our selected diseases is actually caused by a spirochete. The spirochete is Borrelia burgdorferi. Anyone know what disease that causes? It's transmitted by the bite of a tick. Lyme disease. Lyme disease. And that's why for some individuals when they get bit by the tick and it injects this bacteria into them, and the bacteria will swim out from where they were introduced in the bite 360 degrees, so in a circle. And because of that, the immune system will respond, right, and you'll develop a rash. And that rash will be circular, right? And it may even have multiple circles to it. And so it's commonly referred to as a bullseye-shaped rash. And it's very indicative of Lyme disease. And again, it has to do with the fact that the organism will spread out. It will leave the site where it was introduced by this unique motility that it has. Only spirochetes can do that. So you can see a little bit better here the multiple flagella that makes these guys motile and the single one on that one. So what strong cell structure helps prokaryotes hold the specific shapes seen on the previous slides? So here's a hint for you. This structure is believed to play a role in the differences seen between when cells are stained using the Gram stain. The cell wall, right? That peptidoglycan, especially for bacteria, right? But remember, when we talk about prokaryotes, we're talking about Archaia as well, but they also have cell walls. So in general terms, it would be their cell wall. Their cell wall also helps them from osmotic pressure. That's why they have it. Because if you've ever played around with our own cells, right, eukaryotic cells, like for instance red blood cells in biology lab, they don't have a cell wall, right? So if you put them in a hypertonic solution, what happens to them? Hypo, right? They get big, they expand, the water comes rushing in and they can even burst. So a lot of times for prokaryotes, they're living in an environment where the pressure is for water to come in, right? And they would burst if it wasn't for their cell wall, right? It helps them resist that pressure by water, that osmotic pressure that happens in the environment for them. We don't have protection from that, but our bodies in general help protect us, right? They maintain a balance of osmotic pressure so that our cells aren't popping all the time. So these guys, this is all one organism in this picture. What shape would you say they are? They're all different shapes, huh? So what do you think they're lacking if they're not holding a shape? They don't have a cell wall. The term that we use to describe these bacteria that don't have a characteristic shape is pleomorphic. Pleo meaning many, morphic referring to shape. There's one entire genus of bacteria that don't contain cell walls and therefore are pleomorphic. And they are called mycoplasmas. And so I always remember plasma, like plasma membrane. I always think all they've got is a plasma membrane. They don't have a cell wall. Oops, sorry, I apologize for that. I thought I turned it off. So myco is actually the prefix for mold. And as you can see, some of these guys kind of stretch out because of no cell wall. And so they almost look like fungi. They almost look like mold. And so that's probably why they chose the name mycoplasma. So mold-like. Mycoplasma pneumoniae causes a mild form of pneumonia. Yeah, see how they're stretched out? So this almost looks like hyphae that you would see under the microscope for mold, but it's not. So it's kind of mold-like, but it's not a mold. So Mycoplasma pneumoniae is a species of mycoplasma that causes a mild form of pneumonia, commonly referred to as walking pneumonia, because people are up walking around and they don't realize how sick they are. So when I was in college, one of my friends, who was a resident assistant with me, we were doing rounds in the dormitory, we decided to race up 4 flights of stairs. She passed out at the top. Come to find out she had walking pneumonia. So exerting herself like that, and she couldn't breathe because she had that pneumonia, she passed out. So, yeah, that was kind of frightening night, to say the least. The bad news with this, and not so bad if you're allergic to penicillin, so you can't take it anyways, you can't take penicillin against this infection. Anybody know why? What do these cells not have? They don't have a cell wall. Guess what the target of penicillin is? Peptidoglycan. It stops the cells from making an efficient cell wall out of peptidoglycan. If you were to give your patient this, if they had walking pneumonia, it'd have no effect on this bacteria because it doesn't have a cell wall, right? So that it would be useless. And so that's why it's important too, like we've talked about gram stain, right? Who comes out gram positive? What is structurally unique about gram positives? Why did they stay purple? Not. The crystal violet and the iodine get trapped inside by what? What traps them? The peptidoglycan, right? They have a very thick peptidoglycan layer that gets dehydrated by the alcohol, right? And so the gaps in between the molecules get smaller, and the crystal violet iodine, that large complex, gets trapped in. So that's why, like if they do a test, a very simple gram stain, if it comes out gram positive, then they could probably use penicillin to treat that patient, right? As long as that bacteria isn't resistant to penicillin, which unfortunately a lot of bacteria out there are. They have enzymes that can destroy penicillin. But in this case, it wouldn't work because this guy would come out gram negative, right? He has no peptidoglycan to trap in that crystal violet. He's going to come out pink, which is going to tell you that penicillin, not a good idea, right? You have to give something like erythromycin, which targets the ribosomes inside the cell as opposed to the cell wall.
What targets the ribosome?
Erythromycin. So then when we talk about shape, as I said, sometimes they'll stay next to each other. So this shape is coccyx, right? Spherical. Sometimes they'll stay together in pairs. We refer to these as diplococci. Diplo referring to pairs. Sometimes they'll form chains of cocci. That's referred to as streptococcus. And again, another one where it's a genus name as well. So Streptococcus priogens, which is actually a species that causes strep throat. Which is kind of good because I tell myself this little story to help remember strepto versus staphylo. So whenever I say strepto, I always think of strep throat. And I think about how painful your throat is when you have that infection. If you've never had it, everyone else is jealous. Because it's horrible, right? You would definitely never want to swallow a chain either, right? So I think about my throat, the length of my throat. I think about a chain. And I think about how I would not want to swallow that, right? So I always remember strepto, strep throat, a chain, and I usually don't get them confused. Now these guys, on the other hand, this is Bacillus megaterium, so notice they're rods, right? But do you notice the arrangement or the grouping they're forming? They're forming chains. So the term that we would give to this arrangement would be Streptobacillus, which again is a genus name, but it's actually being phased out. Because I found that, again, although the organisms all are bacilli and they form these chains, they're actually quite different from one another. So some of them, a lot of them have been moved out of this genus to other genuses. And Bacillus megaterium, is a bacillus. And it is one of the ones we use in lab. I haven't looked it up, but I have a feeling part of the reason why I got this name, megaterium, is that these are the largest rods we work with. I mean, they're huge under the microscope compared to all the other ones we work with. So I wonder if the mega refers to large. For cocci, we've got so many more arrangements because of them being spherical and how they can arrange in three-dimensional space. So we have ones that can form packets of 8, so they divide perpendicular to each other. This is referred to as Sarcinia, which of course again is another genus name as well. If they divide randomly in three-dimensional space, they tend to form these clusters, sometimes referred to as grape-like clusters, because they look like that. This one is Staphylococcus. Staphylo, again, means clustering or grouping. Also another genus, right? And most of us are aware of Staphylococcus aureus, which can cause pretty serious skin infections. Although most of us carry it in our nose, but when it gets in the skin, it can cause pretty serious infections. So again, when they say somebody has a staph infection, they're abbreviating the name of the organism that causes that infection. It's Staphylococcus aureus. Now you have Staphylococcus epidermidis, which lives in your skin, that usually doesn't contribute to any problems, right? It has a symbiotic relationship with our skin, unlike Staph aureus. When it gets into our skin, it can cause pretty serious problems. And then some of you guys may be aware of some other strains of staph, right? We abbreviate it MRSA. Some people say MRSA, but it's an abbreviation. Anyone know what it stands for? Mictillin-resistant Staphylococcus aureus. Micticillin is an antibiotic similar to penicillin. It's a penicillin derivative. Anything that really ends in cillin is a penicillin that's been chemically modified, right? So amoxicillin, ampicillin, all those other cillins are chemically modified penicillin. So that particular antibiotic was used widely for staph infections. So much so that we have strains now that are resistant to that antibiotic, to basically all penicillin-like antibiotics. They, for the most part, have enzymes that destroy the antibiotic. So the antibiotic goes in and gets destroyed and doesn't do its job. So now, right, we've switched to a different drug. Anyone know which one? It starts with a V. Vancomycin. So guess what we have now? VRSA, vancomycin-resistant Staphylococcus aureus. Will this continue? Oh yeah, it's a scary world we live in, right? And just recently, they found a bacteria that's resistant to all known antibiotics. It's pretty frightening. All right, so we definitely need to discover new ones, right? Which is why I'm going to be gone for a week. I'll learn how to do that. And how to teach my students how to do that. So this is a great summary that I stole from the internet that has all the cocci arrangements. So one of the ones that wasn't shown on the previous slides that I have is this arrangement where they form these square packets of 4. This is referred to as a tetrad because tetrad actually means 4. So diplo, remember, is pairs. Strepto is chains. Tetrad is tetrad, or 4. Sarcinia are those packets of 8, or almost like a cube. And Staphylo is the clusters. So remember, when we talk about prokaryotes, we talk about bacteria and archaea, although we tend to talk mostly about bacteria because it's what we've known the most about for the longest time. You can stain any cell using the gram stain procedure, and you're going to get one of 2 results. It's going to come out purple, which we call gram positive, or it's going to come out pink, which is gram negative. For bacteria, if it comes out gram positive, as we know, it pretty much means that it has a thick peptidoglycan layer. That's what PTG stands for. And we know bacteria have peptidoglycan, right? It's a defining characteristic of that group of organisms. Although there are some that don't have it, like the mycoplasmas, but they're very rare. And because of the mycoplasmas, if it comes out gram negative, it could have no peptidoglycan at all in the case of the mycoplasmas. But for the most part, if it's a gram-negative bacteria, it has a very thin layer of peptidoglycan. And then it also has an additional layer outside that peptidoglycan. Do you guys remember what it's called? So gram-negatives, right, they have a cell membrane. They have a thin layer of peptidoglycan, and then they have another layer. Do you guys remember what that other layer is? The outer membrane. Wow, that one's easy, huh? They weren't very creative in that name. It's very helpful, though, right? You know it's outer, and it's a membrane. So peptidoglycan, as we're going to talk about in a moment, is made up of sugars and amino acids. So that chemical structure, as we know, we believe it shrinks when we expose it to the ethanol. So it makes sense that archaea, which don't contain peptidoglycan, but some of them, when you gram stain them, they come out gram positive. And that's because their cell wall is made of pseudomurine, which is a similar substance to some of the sugar-like substances that some organisms have, or they're made of complex polysaccharides. And of course, polysaccharides are what? Sugars, right? Poly meaning many, saccharide means sugar. So it makes sense that when you expose it to the ethanol, it probably shrinks that cell wall and traps in the crystal violet iodine, hence why they come out purple. Unlike the archaea that come out gram negative, these guys lose the crystal violet iodine when you decolorize. And that's because their cell walls tend to be made out of glycoprotein. This is sugar and protein. But this interaction is such that you don't get that shrinkage. Or they're just made of protein, their cell walls. So not enough sugar, right, with that glycoprotein to create that shrinkage that you get that would trap in the crystal violet iodine. So remember, when we're talking about bacteria, gram-positive versus gram-negative, what do their cell walls look like? And how do these differences contribute to their gram stain reaction? And this is something we went over last time, right? So this is a little bit of a review. How many of you guys have done your Connect assignment for gram stain? Woohoo, yay. If you haven't yet, do it over the weekend as well as Chapter 1, right? And you'll notice the agreement. I asked you guys questions and reminded you some of the things that as it relates to Connect. The other thing is if you want to read over that agreement again, you just click on it again using the lockdown browser and you can read over it again. So knowing, right, what we know about how the gram stain works or how we believe it works, we could hypothesize the outcome of the gram stain reaction for other cell types based on the concepts behind the gram stain and how it works for bacteria. So we kind of already hypothesized for and have some data for the Archaia. Now let's talk about a different situation. So how does the gram stain have anything to do with the charge of the cell? So notice, again, we say gram positive or gram negative. Are we talking about a charge when we say that? No, we're talking about a result in the gram stain reaction. What charge do all cells have? They all have a negative charge. So the dyes that we actually use in this, because they stain the cells, are actually basic, which have what charge? Positive charge. Okay. So remember, this isn't about charges per se, right? When we say gram positive versus gram negative. So what structure is present in the cell walls that causes the different results seen in the gram stain reaction for bacteria? What's the cell wall made out of for bacteria? Peptidoglycan, right. So, how would human cells come out if we were to gram stain them? Would they be gram negative or gram positive? They would be negative. There's two reasons why they can't be gram positive. One, do they have a cell wall? No. So nothing to trap in the crystal violet iodine, right? Well, if they don't have a cell wall, the next one is null and void, right? Because it's definitely not made out of peptidoglycan, is it? So the fact that they don't have a cell wall, and it's certainly not made of peptidoglycan because they don't have it, they would come out negative. They wouldn't trap in the crystal violet iodine. And that's true. Like if you were to do a cheek swab, right, and you put it on a slide and you gram stain it, our cells will come out pink. And the nucleus will sometimes even be a brighter pink, almost red. Because remember, what's in the nucleus that's highly negatively charged that attracts the positive dyes? The DNA, right? The nucleus, that's why it stains so well for eukaryotes. It's all that DNA in there. And the membrane itself is negatively charged, so it usually gets stained really well too, and that's why you can see the membrane, right? Most of the time when you look at cells, especially in anatomy lab. But you might even pick up some bacteria that are normally in your mouth, right? And a lot of those are gram positive. But again, they're going to be tinier than your eukaryotic cells as well. So gram positive versus gram negative. And we were allowed to put pictures and stuff on our office doors. I actually had this one on my office door for the longest time. So again, remember, really thick peptidoglycan layer for our gram positives. If it's present, which it is for most gram negatives with the exception of mycoplasmas, very thin layer peptidoglycan. And usually most gram negative bacteria have that outer membrane. And so we're going to start looking into the specifics of these membranes. So we're going to talk about their composition, what they're made out of, how they function, and some of the limited internal membrane structures that are found in some prokaryotes. So the membrane, and these aren't all that different from our own membranes, right? We know membranes are made out of phospholipids, and they have proteins and other substances embedded in that layer. So therefore, sometimes it's referred to as the fluid mosaic model. Fluid implies that it does what? It moves, right? This is not set. Unlike most mosaics, right, where they break up tiles and glue them on a wall to make a mural, mosaic in this use means that it's made of many parts to make the whole picture, right? That's what they mean by mosaic. So you've got the phospholipids, you've got the many different proteins and protein-associated molecules, but it's fluid. It's constantly changing and moving. So these proteins are almost like icebergs floating through a phospholipid sea. Right? They're continually moving through this very fluid layer. So it's flexible, it's dynamic, it's constantly changing. There are sterol-like molecules embedded in the membrane. These are referred to as haploids for prokaryotes. These provide additional support and rigidity to the membrane. We have sterol-like substances in our membranes that help with rigidity and stability. Does anybody know the name of the molecule in our cell membranes? It's something we actually keep track of in our blood work a lot, because some people overproduce it and some people eat too much of it. Cholesterol. Cholesterol. Animal cells have cholesterol in them naturally. We actually make cholesterol to support our membranes. The problem is that if you eat a lot of animals, you also ingest a lot of cholesterol, right? Which Kat doesn't have to worry about because she's vegan. But she could still have a cholesterol problem. Why could she still have a cholesterol problem? Hereditary, right? Some people genetically, unfortunately, produce too much. Yeah. Right? And so that's why we have medications that can help with that. So yeah, when it comes to cholesterol, it can be a twofold problem. It can be what you're eating, and it could be what your genetics are, right? The good news as far as what you're eating, you can change that. The bad news when it comes to genetics, you can't really change that, right? But you can take medication. And of course, in that case, you definitely would want to watch your diet. So phospholipids are the main component of membranes, right? They're called this because they have a phosphate group, right? And it's commonly denoted by this ball. And then fatty acids, which are hydrocarbons, carbon and hydrogen, a lot of it, and sometimes a little bit of oxygen. This creates a molecule that's what we call polar, because the top portion is charged. You see the plus and the minus. And so you have this pulling of electrons from one end to the other. And so these guys are pulling the oxygens. Oxygen is very greedy when it comes to electrons, right? Just like with water molecules, right? That have that polar structure to them because oxygen is very greedy. He wants all the electrons. And hydrogen isn't as greedy, and so he tends to be positively charged because his electrons are being pulled away from him by oxygen. Go ahead and, I think unfortunately they haven't unlocked our doors. Hello. So we have to remember not to close that when we leave, or we'll be locked out. I'll have to call campus police. So you have this positive minus, and water is like that too. So water likes molecules that are charged, right? Because they're charged as well. And so they interact with each other really well. So this part of the molecule is referred to as being hydrophilic. Hydro referring to, again, is water. What does philic mean? Loving. Unlike this end is not charged. The fatty acids are not charged. So they're nonpolar, right? You don't have this charge. You don't have this pull. Everybody's good at sharing. Like carbon and hydrogen are really good at sharing electrons. So in this case, that part of the molecule, because it's not charged, tends to be hydrophobic, which means fearing water or hates water. Phobic means fear, right? So these molecules naturally in nature, if you just stick them in water, they form what's called micelles. And they'll point all their phosphate heads out, and all the fatty acids will be on the inside. And what happens when you mix oil and water, right? The same thing happens. When you make your own homemade salad dressing, right? Vinegar is very similar to water. And oil, you cannot truly make those guys go together, right? You create an emulsion when you shake that up. You make little tiny fat droplets that are really unhappy because they're surrounded by water, like molecules. So when they have the chance, what do? they run back to their buddies and they separate. So you have that oil layer and that vinegar layer, water-like layer, right? So we run into the same problem with digestion, right? And that's why we produce bile salts in our bile that gets added into our small intestines. And this is to help break up those little fat molecules so that we can actually absorb them, right? The bile salts create a barrier between the water in our intestines and the fats that we ingest. And that's why when people go on crash diets and they don't eat fat for a long period of time, the bile does not leave the liver, and you're actually always producing it in your liver. But it goes down the bile ducts, and it goes back up if that little sphincter in your small intestine is closed. And so then it backs up into your gallbladder. If it stays there for a long period of time and isn't squeezed out, and your gallbladder is just literally like a little bag that you can occasionally squeeze and get extra bile to go into your small intestines. And of course that's going to happen when you eat a really fatty meal. Like if you live in New Orleans, that's pretty much all the time, right? You go have a shrimp po' boy, right? Your gallbladder squeeze it, trust me, right? And shooting some extra bile into there, into your small intestines to help with the digestion of all that fat you just ingested. If you don't use it, then crystals can build up. And then when you actually go to use it, the crystals get trapped and damage the duct. So then you have people who have had their gallbladder removed, right? Because it's been damaged by those salt crystals that build up because they didn't use it for a while. So a little bit of fat is a good thing, right? Because it'll keep getting the stuff out of there, and it won't build up. So anyone who's had that surgery is never going to eat a shrimp po' boy again, because guess what? You don't have that extra little bag of bile to squeeze. So your liver can't produce enough quickly enough when you eat a fatty meal like that. So it just goes right out. And no more shrimp po' boys for you. So, once a week, it's good. It's all good. I can't eat it anymore. It's freaking flour on the breading of the shrimp. They need to make grilled shrimp po' boys. I tell Parkway they need to make grilled shrimp po' boys, right? I can't eat the bread either. I just go and get the beef by itself, but the gravy's probably got wheat in it too, but I just suffer. Okay, so it's a horrible wheat world. So fats, as we said, right? Carbon and hydrogen. So in this picture, right, carbon, as many hydrogens as they could possibly have here or there. So this molecule is said to be saturated. Carbon can make 4 bonds with 4 different molecules. So notice here, carbon is binding with 3 hydrogens and the carbon next to it. Here, these carbons are bonding with each other and 2 hydrogens. So this guy is packed, right? So this is what we call saturated. And these fats are what at room temperature? These are solid at room temperature, right? That's the clue. Or it's been hydrogenated, which means they've added hydrogen to make it a saturated fat. They do this with things like peanut butter, right? If you don't have to stir your peanut butter, if you don't have a liquid layer of your peanut butter, then it's been hydrogenated, which means they added hydrogen to it to make it saturated so it would not be oily. Yeah, you are supposed to have it stirred in. It's the unsaturated fat, that's why it's oily, right? So unsaturated fats don't have the maximum amount of hydrogen like you see in the next picture and will be liquid at room temperature. So these guys, they're not packed, and so in several places they'll tend to have double bonding, double sharing between the carbons. But what this does is give the molecule more flexibility, hence why it can be fluid at room temperature. But notice I'm saying room temperature. Does temperature affect? Yeah. So if you take a saturated fat, like butter, can you make it liquid? What do? You heat it up. You heat it up. So then you affect the bonding, you affect its fluidity. Can you take a saturated fat and make it saturated? Yeah, you can add hydrogen to it, right? Do evil things like that. But for the most part, you can stick oil in the freezer and it's not going to freeze. Yeah. It's going to stay flexible. Really.
Like, bro, really? I didn't know that. That's crazy. That
Yeah.
blew my mind a little bit.
Yeah. Well, it's all chemistry. It's all chemistry. That's all cooking is too, is chemistry. It's cooking science. So this is what membranes are made out of, right? Out of these fatty substances. That hydrophobic tail. So do you think that organisms have membranes that are saturated or unsaturated? You think they're saturated? What would justify that choice for you? So what do we know about saturated? It's solid at room temperature, right? So the membranes would be pretty rigid, right? Do you want a rigid membrane? Well, if you live in a hot environment, though, you might need that rigidity, right? So it would really depend on the environment you live in. So if you live in a warm environment, you probably need those saturated fats so you can stay a little bit more rigid. If you live in a cold environment, which one do you think you'd want? Unsaturated, because it could still move, right, under those cold conditions. Make sense? So Archaea have modifications to their membranes, because remember, when we talk about Archaea, there are extremists, right? They're the ones that can live in the extremely salty and extremely cold and extremely hot environments. So some of the adaptations they have is their fatty acids are branched. So notice it's not a single zigzag line, but you see these little spikes coming out, right? That's the branches. So again, this is a much bigger fatty acid. So not as much mobility, which would make sense too if you're in a really hot environment. You don't want to fall apart. You want to stick together. The other difference is the ether bond instead of the ester bond that you typically see. So what the heck does that mean, right? I just terrified you with organic chemistry, didn't I? Terrified myself because I can't remember one from the other. So do I expect you to? No. The important thing I want you guys to know is they're different. And what they're talking about here is this bonding here between the phosphate group and the fatty acids in this glycerol component. This is what's referred to as an ester molecule, where it's a different structure, it's ether. And I'd have to look up my organic chemistry to tell you the exact differences. I don't think you'd remember it either. The important thing is it's different. And why is it different? Because these organisms live in extreme environments, in different environments than bacteria. They can also form monolayers. So typically when we look at a membrane, it's two layers, right? It's what we call a bilayer. These guys can have it connected, right? So it's all one layer. They can even, and these blue things are to represent embedded proteins, by the way. They can also cause their carbon atoms to form cyclic bonds. So these little houses that you see here, these, it's 5 carbons. What is 5? Pentose, right? So these little pentose rings you see, that when they ring up like that, notice it shortens the membrane. So they actually even have the ability to change the width, the diameter of their membranes. And again, these are adaptations that help them survive in the extreme environments that they live in. Think about this. If your two layers are stuck together, are they likely to come apart? No. So like if you're in a really hot environment, if you're a monolayer, you're most likely going to stay together, right? Then come apart. The whole shrinking thing, I don't know why they do that. But, it's, again, probably an adaptation to the extreme environments. So, what's the function of membranes? Hold the stuff in, right? it's semi-permeable, right? It defines the cell, and it is a barrier, right? It's like the picky bouncer. He decides who's going to get in the nightclub, right? Not everybody gets in. You got to bribe them or be pretty. I bribe. Right? So those membranes, right, really control what happens. And cellular processes happen in those membranes. Do you guys remember from biology class what proteins like cytochrome C and things like that, those proteins are embedded in a membrane. They're part of a very important chain reaction that happens in the membrane. Yep, you said it. Electron transport chain. Remember that? Where electrons that we take from things like glucose get carried by electron carriers like NAD and FADH, and they give those to electron carriers. Those are embedded in a membrane. And those shoot hydrogen ions, so a hydrogen atom that's lost its electron, right? So it's a proton, across that membrane, and then when they flow back to the other side, they kind of go through a turbine, right? A protein that drives the force and the energy to add phosphates to ADP to form ATP, right? So metabolic processes like electron transport chains happen in the membrane. So if you're a bacterial cell, this happens in your membrane, right? Is the cell membrane enough area for that to happen? Sometimes that's not quite enough. So as you see in this picture, you'll see folded membranes inside the cell. You see these lines? You see these lines? See how they're folded and stacked? Does look like an organelle to you guys that some eukaryotes have? Endoplasmic reticulum, or plants. Plants have stacked membranes in one of their organelles. Chloroplasts. And there is the belief that mitochondria and chloroplasts were once free-living prokaryotic cells engulfed by a pre-eukaryotic cell. And they began this symbiotic relationship so much so they can't live without each other. But that, again, these membranes, those cellular processes are happening there. That electron transport chain, that's what's happening there. And that's why they have them inside their cells. Now, it's not organized to the organelle level like we talk about with organelles in eukaryotes, right? This is a prokaryotic cell. But you'll find prokaryotic cells with stacks of membranes inside of them. So it's not an organelle, right? But there are internal membranes. Because basically what you're looking at is an organism very similar to organelles we find, mitochondria, chloroplasts. So they can do it all on their own, right? They're a single-celled organism. So you need to be able to identify the major prokaryotic structures in a drawing or a micrograph for prokaryotes. So general appearance, what would these things look like? What's their composition? What are they made out of? And how do they function? So we're going to look at inclusion bodies, ribosomes, the nucleoid, plasmids, and the eukaryotic cytoskeleton. So we're actually going to talk about these in our next lecture. And typically what I would have you guys do in preparation would be to fill in this table. But we'll do it together as we go to the next lecture. So we get to take our break now. So we'll come back at like in 15 minutes, so at 7:33.